Hello everyone, welcome to the channel KR Microbiology classes. In this video, today we will discuss about the industrial production of ethanol, which is also called as the alcohol. So, in this industrial production of alcohol, we will learn about the fermentation process, recovery, strains, media, whatever we are using for the production of ethyl alcohol, we will discuss. So, ethyl alcohol or ethanol, it is commonly called as the alcohol and it is volatile in nature. And if you see this ethanol, it is useful in so many areas like pharmaceutical industries or in medical field or in cosmetics, something like that. Okay, and actually ethanol production in the very starting time in 1930s, like people used to produce this ethyl alcohol through fermentation process, but later they started to produce through the process of dehydration of ethylene. Later, this ethylene, getting this ethylene became a very expensive thing, like crude oil prices were increased, and it is also one of the non-renewable resource, no, crude oil. So that's why again people turn to produce the produce the ethanol through the process of fermentation. Later, if you see what kind of microorganisms we are using for the production of ethanol in the fermentation. So if you see here, yeast. In the yeast, Saccharomyces cerevisiae. So this is the very important microorganism using for the production of the ethyl alcohol. So it is commonly used by everywhere. And Saccharomyces uverum. So it stood in the second place. After the Saccharomyces cerevisiae, mostly these industries will use the Saccharomyces uverum. And it depends upon the kind of substrate we use the microorganisms for the fermentation. Like if you see this Candida utilis, it is used to produce the ethanol from the waste sulfate liquor. And if you see this Cluveromyces fragilis, it is used when we use the whey to produce the ethanol. Like this depends upon the substratum. The microorganism we are using for the fermentation will also change. And apart from the yeast, we also use bacteria. It is called as the Zymomonas mobilis. Okay, and later if we see the type of culture media we are using for the fermentation. Here we have to understand that there are three types of media we use in the fermentation of the ethanol. So these three types of media, one is mainly starch containing substrate, and another one is sugar containing substrate, and the third one is cellulose based substrate. If you see the starch based substrate, examples are corn, wheat, sweet potatoes, milo, wheat, rice, cassava. Like all these are the starch based culture media. And if you see the sugar based culture media, sugarcane, beetroot, sweet sorghum, fruits, all these comes under the sugar based media. And if you see the cellulose based media, wood, paper waste, crop waste, wood waste, all these comes under the cellulose based culture media. So depends upon the culture media we are using, we will use the microorganism or we use the culture. Okay, and if you see the conditions, so pH must be 5 and temperature must be 35 degrees centigrade, and the kind of fermentation, it might be continuous fermentation or it might be batch fermentation, whatever it is, okay. And within 12 hours of the when we start the fermentation, within 12 hours the production will start. In the case of Saccharomyces cerevisiae, it will produce 10 volume per 10 to 20 grams of dry cell weight. Okay, and after starting the production, so whenever we are removing the ethanol, the yeast cells are separated and they are used as the single cell protein, and the supernatant is processed for the ethanol. This is the process what we did for the fermentation. If you see the process of fermentation, from the glucose, acetaldehyde will form, and from the acetaldehyde, ethanol is produced. But in the case of starch substrate, first we have to add amylases or cellulases. In the case of cellulose material, we have to add cellulases, or in the case of starch, we have to add amylases. Thereafter, the remaining process will start. That is the formation of glucose from the glucose acetaldehyde, and thereafter final product of ethanol will produced. So this is the process of fermentation for the ethanol production. So after the fermentation process, the very next process is the recovery. So 95% of ethyl alcohol is recovered through the process called successive distillation, and we can also recover 100% ethyl alcohol using the azeotropic mixture. You know very well that azeotropic mixture is a mixture of two or more liquids having similar boiling points and composition in vapor state. So using such azeotropic mixture, we can recover 100% ethyl alcohol. So the next one is the uses of ethanol. So ethanol has a huge applications in so many areas like manufacturing of drugs, plastics, liquors, polishes, plasticizers, cosmetics. Like in so many areas, we will use ethanol. Especially in ancient days, ethanol is used as a sedative in surgery, and it is also used in pharmaceutical preparations, and it is used as a topical agent to prevent skin infections, and it is used as antiseptic disinfectant also. And nowadays we will see, if you see in these COVID time, we are using hand sanitizers, no? So in that hand sanitizer also, if you observe, they will mention like 70% ethyl alcohol, 90% ethyl alcohol, like that. So this ethyl alcohol or ethanol is used in the hand sanitizers like a antibacterial and antifungal component. Yeah, so this is all about the ethanol, its production, fermentation, conditions, and its uses, recovery, and everything. Thanks for watching this video. If you like this video, please like, share, and subscribe. Thank you very much. Thank you, Nana.